The process of double fertilization is a unique reproductive mechanism found in flowering plants. When pollen lands on the stigma of a flower, it begins a complex fertilization process. A pollen grain containing two sperm cells lands on the receptive stigma. After landing, the pollen grain germinates and forms a pollen tube that grows down through the style toward the ovary. The pollen tube carries two sperm cells down to the ovule. Once the pollen tube reaches the ovule, it enters the embryo sac, where the egg cell and two polar nuclei are located. The first sperm cell moves toward the egg cell and fertilizes it, forming a diploid zygote that will develop into the embryo. Simultaneously, the second sperm cell fuses with the two polar nuclei, forming a triploid cell that will develop into the endosperm, which provides nutrition for the developing embryo. This unique double fertilization process is what distinguishes flowering plants from other plant groups. Let's explore the significance and outcomes of double fertilization in flowering plants. Double fertilization is evolutionarily significant because it is unique to flowering plants, or angiosperms. The first fertilization produces an embryo that will develop into the new plant. The second fertilization creates endosperm, a nutritive tissue that provides food for the developing embryo. This endosperm is why seeds like corn, wheat, and rice are nutritionally valuable to humans and animals. Double fertilization ensures that plants only develop seeds when fertilization occurs, conserving valuable energy and resources. After double fertilization, the ovule develops into a seed, while the ovary develops into a fruit. These processes complete the reproductive cycle of flowering plants, from flower to seed and eventually to a new plant. Thus, double fertilization ensures reproductive success and continuation of flowering plant species.